Hi, everyone. I hope, well, I see myself, so I hope you can also hear me. Well, I don't hear you, so I'll just assume it's
Again, you
okay.
need to talk to her. Diz que sim. Respond yes.
Okay.
Yes, yes, yes.
All good?
Yes. Yes,
Okay,
please,
thank
go
you.
ahead.
Yeah. All right. So, yeah, thank you very much for inviting me and for getting over there to watch my talk. So, today I want to tell you about the work that my group is doing at AMBL for image segmentation of microscopy data and how we are trying to incorporate the human knowledge about the expectation of the things that we will find in microscopy images into the segmentation procedures. Right. So, segmentation is a well-studied problem in computer vision. Well, it's well studied, but it's not actually solved yet. And there are many flavors of the segmentation problem. And the particular one we want to look at today is instance segmentation. Right? So we want to extract all the objects out of the image into like separate labeled masks. Right? And this problem is particularly important for microscopy because essentially this is why you are doing the imaging. Right? So you want to image biological objects and then extract them and perform some measurements on them and formulate your biological hypothesis, maybe test it with the next experiment. So you really want to like find all these objects separately and then measure what is happening. And that's why you need segmentation, which can be easy. Like in the case I'm showing here, this is something we can easily do with conventional methods, with like random forest-like machine learning. It can also be more difficult, especially if you are trying to segment, like say volumes, especially in live imaging, when you have to make a trade-off between keeping your sample alive for long enough but having good image quality, or if you have to make compromises because of the speed of imaging and the resolution. Right, so this is already getting a lot more tricky. And then finally, there are also electron microscopy segmentation problems, which are even more tricky because the staining in there is not selective. Like, in light microscopy, you can label exactly what you want to see. Here you can't, and here everything gets labeled. And then you have to figure out which of these structures that are labeled are actually the ones that you're looking for. So yeah, that's the kind of problem that my group is actually interested in. Especially the segmentation problem when you have to go from the boundary information, like in the one that I just showed, right? So you see here the boundaries are labeled, and then this is where the objects are. And then also in electron microscopy, as you will see later, this is also a boundary-based segmentation problem. And the way people usually approach that is, so this is like the standard established segmentation baseline, and I wanted to show it to you so that you would see then how we can improve that by introducing additional prior which all belong together. And then these superpixels you agglomerate to actually create the final objects that you're interested in. And well, this works well, but it has its limitations. And the limitations mostly come from the problems of, well, there are sample preparation issues, where you see like some of the arrows on this image are pointing to areas where we think there should be a boundary, but there isn't one, right? So it's missing in the image. We don't know why it's gone. It happens. Right? So sometimes parts of your boundaries will just not be there. Another problem is that you also, especially in a very large sample, like say if you're doing whole organism imaging or tissue imaging, then there is a lot of biological variability. So all these cells are of different types. They all look different. Their membranes also look kind of different. And well, that's a problem because if you want to predict these boundaries, and we are following the regular, totally normal supervised machine learning paradigm, you need a lot of training data to actually represent all this variability. And we can't be like the natural image people who would say, well, there is ImageNet, right? We'll pre-train everything. We'll have a backbone. Other people will do it for us. So here, we would have to collect the training data for all the possible ways these membranes or like separators of cells can look like. And this is not feasible because collecting this training data is extremely expensive. You have to have professional biologists who would sit down and actually paint on these images in 3D. It takes months of time. It takes tens of thousands of euros to create enough training data to even get to the state where we are now. Right? So creating more is just not possible. And this is why we actually started thinking, like, how can we then replace this part of that could be easily solved with more training data, but like I said, is not possible, by saying that, well, we know that some things in these images, we know what they are, we know which parts should be together or not. So what can we do about this? And here, yeah, so if we think of this, the general pipeline, right, in the boundary prediction, there is not much prior information that we can introduce because, well, boundary prediction is just learned. And the boundary appearance, it would be hard to encode what we expect of it out of just, besides just labeling them. So we started looking at the second step, which is this graph-based agglomeration, because here we are actually having like the objects in the end, and on the objects we can have priors, right? So let's look more closely what's happening there. And if we have these superpixels, and so you connect all these superpixels into this superpixel region adjacency graph, right? And then the way the graph agglomeration works is then you say, okay, some of these edges are very strong, they want to stay on, they have high probability of the boundary, for example, associated with them, and some are very weak and just came out of the watershed because, that's where the waterfronts met. Those are weak and can probably go. And now imagine that on top of that, on top of these edges that you have now estimated like this, you could also say, you know from some oracle, right? You know from biology that there are two locations in this image which should be separated, right? Then you say, okay, so if this should be separated, you can introduce an additional edge in this graph and say then that this should also be separated. And well, more formally, but still not very formal, if you look at the graphs like this, right? So if you just have a graph, like I'm showing on the left, and this graph it's a region adjacency graph, it has positive and negative edges, where the positive edges are the ones that really want to stay on, right? These are very strong edges, very high boundary probability estimated before. And then you have the weak edges that can probably go. And finding the right balance between these edges is known as the multi-cut graph partitioning problem. And the group of Bjorn Andres has been introducing different ways of how to solve it and how to enlarge it over the last years. And the contribution that we have made there of connecting it to the prior knowledge was to introduce the edges which are connecting something that is not close, but they are sparse. Right? So you can select two locations in the graph and put like one edge between them, but then say that, okay, these two locations should really be separated. Or find a way of how they should be merged. I'm sure they actually belong together. So what can we do with these kind of things? So what kind of priors can we encode in an image like we have from the electron microscopy? Right, so here you can see the raw data, right? And on the right, these are the superpixels that we are working on. And then, well, we know, for example, that we can segment nuclei in these images. And nuclei are, because the chromatin texture looks very different from everything else, nuclei are actually fairly easy to segment. So we can segment the nuclei, and then we can remember our biology and say that, well, every cell should only have one nucleus. So then you can introduce this kind of forces that go away and push all the other nuclei away. Right? And in the same way, you can then say, okay, and then if things belong to the same nucleus, they should actually be together. Like other forces you can introduce, you can say, well, if you have a tissue boundary, then obviously a cell should not belong to two tissues at the same time. So then you can detect the tissue boundaries and put repulsions to everything outside. And then if you have a horrible segmentation error, like the one that you see there, where some boundary was not detected and it just overspilled and overmerged into a catastrophic merge, right? And then we can put some tissue boundaries on top that we can easily predict from a very downsampled image, right? And then it can actually become a nice segmentation. So using this and a couple of other rules like that really push things together, push things apart when they should be apart, right? We achieved the full segmentation of this 8 terabyte volume of a whole marine worm, 11,500 cells. And then with that, you can also get the full segmentation of nuclei and the segmentation of the chromatin domains in the nuclei, and also a few other things like the animal cuticle. And well, then it turned out that, well, having segmentation like this is actually a very enabling technology, and our colleagues in biology have used it then to build the whole resource of how you can analyze an animal like this and analyze the morphology of every cell, also together with the gene expression information. So that was fun, but it felt like we should be able to go much further, because this kind of repulsive and attractive potential, that's like a nice abstraction, but there is actually more. I mean, biology is full of different patterns. And we should be able to exploit these patterns to make our segmentations better and not to rely so much on the training data that has to be produced again and again for each new data set that we have to segment. And the particular kind of thing that we wanted to exploit is the concept of stereotypic development. So what you see in this movie is the development of a Drosophila embryo, which is a very, very well studied model organism. And basically, developmental biologists know everything about how this development happens. So for every cell in there, you know when it will divide. You know approximately where it will go. You know that gastrulation will start and some of them will get in and then they will separate into this segments in there. So we know a lot. And we really want to be able to actually exploit this. And this is how we have set out on the next project to incorporate this kind of biological priors, including the non-differentiable priors and different kind of measures. And we also want to do it directly in the loss and not in this kind of a post-processing step. So this is the first variant of it is on archive, but we are also still very actively working on this with more ideas. Yeah, so how exactly can we do this? If you look at the pipeline there, right, like I said, we have the boundaries predicted, superpixels, full segmentation, and well, you have to remember that the priors that we have are on the object level, they're not for boundaries. But this step it's not learned, and in any case, the objective of partitioning the graph is not differentiable. So if we look at it in more detail again, right, it's an NP-hard discrete optimization problem. They're good, very good and efficient approximate solvers. As an input, it takes the weights of the edges, and the output of this is a consistent segmentation that doesn't have dangling edges. Right, so the idea that we had was then to use reinforcement learning and then take everything that is not differentiable and put it into the environment. And then we have an agent that would predict the weights of the edges, right? And then out of the weights of the edges, we would then look at the segmentation and using whatever priors we come up with, say, look at the object, see how much they conform to our priors, and then return that as a reward to the agent and hope that it will then do better. Right, so as a full setup, this is what we have. Right, so we have the agent in there. So the whole pipeline starts from having superpixels, right? And then on the superpixels, we have some features of the superpixels like intensity. I mean, they are actually learned in a self-supervised manner. So let's just say they're provided, right? These superpixels and the features, they comprise the state. The state goes into the agent. The agent looks at the graph with the superpixels and decides what weights it wants to put to the edges. Right? And then in the environment, we actually create the graph partitioning, generate the reward by looking at how much these objects are close to what we expect them to be. And that goes back to the agent that then tries to do better. Yeah, so what can you actually put into these rewards? Well, whatever you want, right? So we want to be able to put anything, for example, size. Like in biology, we very often know the expected size, the number of objects, their shape, orientation, or position, the granularity of the priors that we want to be able to put in is, well, naturally it's per object. There are also things we expect per image. Like I said, for example, the number of objects. We can also do per subgraph, right? And then the first experiment that we wanted to try it on was a very synthetic image like this, which is actually very hard for boundary-based segmentation methods because the background is so structured and has so many edges. So out of this, we can make superpixels, which have edges, right? And then we generated this kind of a reward surface, which would look at the half transform, which basically measures the circularity of your objects, and then the total number of objects, and we balance it out, right? So if we have, for example, if we have not enough objects, then we try to make more. If we have too many, we only take the ones that are very circular. Right? You can also make it more formal, but let's not stop on the formulas now. Yeah, so then you can see already in here, if you look at the actions where the red edges are the ones that it really wants to become very strong, right? And then in the rewards, you can see that the bright green ones are the ones where it has very high reward. So at the objects which are already correct, the rewards are high. And then in the top right corner where the segmentation is still far from perfect, it actually says that, no, you have to drop all those edges. So we can get a prediction which looks very reasonable just out of knowing that we want to extract circles. And this is much better than the baseline that you can see in the lower right corner, which is just doing it based on edges. Right, so it was not so easy to make it work as we expected in the beginning. And in the end, the setup that we have selected was to do there's this model called the Stateless actor Critic. It has, from what we know, it has not really been used in practice. It's been introduced by Pfau and Vinyals in their seminal work on finding a connection between the actor Critic and the gun world. And the idea there is that there is no sequence. We predict the whole graph at the same time. And then the actor predicts the actions, which are the weights of every edge. And then the critic predicts the action values, given the state and the actions. So what actually happens in there is that the critic, in this case, becomes in a way like a surrogate approximator for these non-differentiable rewards that we have composed out of our prior knowledge. Right, and then to make it work, we found that, we really have to figure out how to make it work on subgraphs because the whole image rewards are not granular enough. Object level rewards are too noisy. So you need to be able to extract subgraphs, which we do like at different sizes. They should overlap so that you have a smooth reward and they should fully cover the original graph. So it's not a very difficult problem. And here you can see an example of all these subgraphs of size 3 in this kind of two-object thing. To kind of, summarize it again, this is the full setup. We start with superpixels, then we compute in a self-supervised manner, we compute the features, and then we go on like this. So then we thought, okay, what if we can extend it even more? So what if we just take the superpixels, no features, nothing. We don't know anything about the superpixels now, rather than their location, and then this accumulation of superpixel features we also put into the agent. So we can also do that. And that we actually needed to process the real life data set that we wanted to work with. So this is this Drosophila embryo that I was showing to you before. This is a cross section. And here you can see there is this really interesting pattern of how all these cells align on this kind of a radial manner around the cavity, which is empty. And here we used for edge prediction, this is an edge detector that was developed for a totally different problem, different microscope, different everything. And so we have the raw data, and we have the edge prediction that comes of that. And and these would be like the features that we can then either define by ourselves, like the size, or we actually have the agent learn them. And then, interestingly, you can see that if you learn long enough, so if you look at this image C, it actually comes to a sort of like a semantic segmentation of the boundaries. And the particular reward that we are doing there is like in the background area, we have a strong reward for merging. In the foreground area, we have the size and aspect ratio of the objects that we know and the orientation in which they should really kind of fetch the image like this. And then to know where the foreground and the background is, we know the width of the embryo, and we just put like a Gaussian ring on top to estimate that. And that's the results that we can get out of this. So you see that the superpixels are still kind of messy, so it's not a trivial problem at all. And then you can see where the rewards are, and then indeed that the actions and the actions and the rewards together really make it come into this kind of a pattern. And then you can also add more supervision, So you can say, you don't have to have a non-differentiable function. You can also put, say, dice, You project the dice on subgraphs. It works even better, because if you have supervision so good. The reason why I wanted to show it to you is that you can see that in this case, it really converges for these PCA features to the fully semantic segmentation of the circles. And then you can also have mixed supervision where you say, okay, let's have just one image labeled, and for the rest, we only give priors. That also works, and it works better than fully unsupervised. To summarize the whole thing, so we first found a way of how you can add this sparse, repulsive, or attractive knowledge to your segmentation. This can be absorbed in post-processing. And then we decided to take another step and actually have it end-to-end trained, also for non-differentiable losses, and which would allow us to incorporate a wide array of priors that we know from biology about the shape, the size, anything you can think of. And so we think we are only in beginning in there, because we can also see that semi-supervised will probably be even better. And the reason why I find this work like I'm happy to work on this is that I find it very inspiring of how you can go from this superpixel image, which is all just little pieces of objects, and then without any other knowledge than the size and the approximate shape, and that they should be aligned in a certain pattern, you can actually extract the objects exactly as they are found in biology. So that's what I wanted to show you today, and I'd be happy to discuss more if you have questions. Thank you.